So my task is now to enlighten you a little bit about the mechanism of the disease. We have some good information based both on our patients and also on mouse models, but we don't know everything yet. So there are two major target antigens in bullous pemphigoid, as most of you know, being a dermatologist. And the first one, the major one, is really BP180, and the other name is type 17 collagen. And this is a transmembrane protein here that is reaching in the lamina densa, then kinking back to the lamina lucida. And most patients, I would say more than 95% of the patients, have IgG antibodies against this molecule, and most of them, about 80%, 75 to 80%, against this non-collagenous 16A domain, the 16th non-collagenous domain here. And about 40%, we will see that, also have IgE antibodies against BP180 and C16A. And then we have a second molecule. This is BP230, bullous pemphigoid antigen 230. They're called 180 and 230 because they're 180 kilodaltons and 230 kilodaltons. And this BP230 is intracellular molecule. It's a plaquine molecule. And here about half of the patients have IgG antibodies, and mostly in addition to BP180 antibodies. And there are about few patients, I would say, let's say 2, 3, 4, 5% that only have BP230 reactivity. Okay, and these antibodies, they bind, of course, to these target antigens, and they are localized here in the hemidesmosome and the dermal epidermal junction. And this is a study we have done a couple of years ago, but similar results have been published by others as well. And here we show that autoantibody levels, they correlate with disease activity. And that is true for both IgG, which is here. This in intravidual patients. So one patient has higher autoantibody levels, there's more disease, and then he gets better, she gets better, then the autoantibodies go down. And this is also true, interestingly, for IgE, shown here, and that correlates with the BPDAI, that is this clinical score for disease activity. So we have really physiological evidence that these antibodies, as they correlate with disease activity, are pathogenic. And that is also true. It has been shown in several mouse models for bullous pemphigoid, especially with IgG antibodies against the murine homologue of the NC16A domain. But we also have one study showing that for IgE antibodies. So without antibodies, you don't have bullous pemphigoid. But now let's go back a little bit to our immunology textbooks about innate immunity. And mostly innate, but we have a little bit adaptive as well. And they can be broadly categorized, these inflammation subtypes, in 1, 2, and 3. Of course, there are stereotypes, and bullous pemphigoid, as you will see, it's not only type 2, which is mainly bullous pemphigoid, but we also have elements of type 1 and 3. So type 1 really is macrophages, natural killer cells, and Th1. For example, you have interleukin, you have interferon gamma. Type 2, that the clinical and also immunology hallmarks are Th2 cells, mast cells, eosinophils, and then you have the key cytokines here, interleukin 4, 5, and 13. And then type 3, more the Th17 type from the T cell. And you also have neutrophils. And there, of course, you have interleukin 17, interleukin 23, and interleukin 22, which is more important, like, for example, for psoriasis. But this is a mixture here. And in bullous pemphigoid, we mainly have type 2 inflammatory diseases. So what are these cytokines doing? We know from interleukin-4, this is, for example, this is in general, so they're important for the differentiation of Th cells to the Th2 line, and also to advance the expression of interleukin-31, which is the main itch cytokine, as you may know from prurigo. And both cytokines really are relevant to recruit inflammatory cells to the skin, and also to stimulate. This is in particular interleukin-4 that is important for autoantibody production and also for autoantibody class switching. And these three cytokines here, they together may play a role in bullous pemphigoid. We will see that in a minute in itch. So what contributes to itch? We have a good idea from in studies from patients' skin, and so we know the inflammation. We have Tier 2 cells, we have eosinophils that are there. We have elevated levels of interleukin-31, interleukin-4, interleukin-13, and they all together, they then stimulate the nerve and then causes itch. But we don't know the sequence of events. We don't know really what is the most important one, and that we need to study, I think, really in vivo in our patients and see if we block one of these, that itch is reduced or not. So to give you a little bit an overview now, what will happen of the sequence of events. So the first thing, of course, is the autoantibodies. They bind to the basal membrane zone here. And then we have different mechanisms. One is very short and immediately, for example, you have the internalization of BP180 that also weakens this dermal epidermal structure, at least in vitro. Then we have the relief of interleukin-6 and 8. We don't know how relevant that is, but this you can see in vitro. And then we have also these autoantibodies not only bind to the basal membrane zone here, they also bind to mast cells. And mast cells, they release a lot of these inflammatory cytokines. And after these three things, a very, very important step comes, it's the activation of complement. Okay, that has at least been shown in all the mouse models that we have. Without complement, the disease activity is significantly reduced. And complement is also then responsible to further attract and recruit inflammatory cells to the skin. So mast cells are already there, as you know, and the cells that come are mainly eosinophils, neutrophils, and then there are a little bit of basophils and also some macrophages. And then you have this Th2, this type 2 inflammatory milieu, where you have mainly interleukin-4, interleukin-5, interleukin-13, and also interleukin-31. And there are more of these cells attracted, and they really bind to the dermal epidermal junction. If you see here, it's not shown, you can read, they line up at the dermal epidermal junction, then they release certain proteases, and that are already well known and reactive oxygen species. And by these proteases, the dermal epidermal junction is split. The proteases, they cleave BP180, but also all the other proteins that are there and hold the epidermis onto the dermis, and then we have the blister formation. And we now have prepared a movie that basically repeats what I've said, but it shows it very nicely with additional pictures and maybe with also a very elegant word.
Bullous pemphigoid, or BP, is a chronic relapsing autoimmune skin disease characterized by itch, inflammatory lesions, and blister formation. In healthy skin, keratinocyte attachment to the dermal epidermal junction is maintained by specialized hemidesmosomal proteins, including BP180 and BP230. Patients with BP lose immune tolerance and develop autoantibodies to these proteins. Consequently, antigen-presenting cells process and display peptides of BP180 and BP230 to naive T cells in the lymph node, which subsequently stimulates autoreactive T cell proliferation. Activated T cells differentiate primarily into Th2 cells, driven by cytokines such as IL-4, and are the main pathogenic subset. Th17 and Th1 cells are also involved in this process. Th2 cells produce type 2 inflammatory cytokines, such as IL-4 and IL-13. IL-4 and IL-13, particularly IL-4, stimulate B cell proliferation, autoantibody production, and class switching to IgE. IgG and IgE autoantibodies targeting BP180 and BP230 are produced, which, upon binding, trigger inflammation through both complement-dependent and complement-independent pathways and the internalization of BP180. Independent from complement, IgG4 and IgE autoantibodies cause keratinocytes to release inflammatory cytokines. The binding of autoantibodies, particularly IgG1, to BP180 and BP230 initiates the complement cascade, which leads to the recruitment and activation of various inflammatory cells. IL-4 and IL-13 not only mediate tissue inflammation, but also, along with IL-31, intense pruritus, a hallmark symptom of BP. This results in sensitization and stimulation of sensory neurons. The interplay between autoimmunity and predominantly type 2 inflammation, along with proteolytic enzymes, results in the development of subepidermal blisters in bullous pemphigoid.